Hello and welcome to another lecture of the course Aquaculture. Today, within the major topic of culture method of tiger prawn Penaeus monodon, we are going to discuss about the biology of Penaeus monodon. Penaeus monodon, we all know that is popularly known as tiger prawn, giant tiger prawn, sometimes black tiger prawn, and about its distribution and systematic position, we have discussed in my previous lecture also that this is widely distributed in tropical to temperate region. This is found in the Indian Ocean and Western Pacific, Indo-West Pacific region, and its distribution we have already discussed is quite wide in the countries of the Indo-Pacific. Morphology also we have talked little bit in my previous lecture about how it looks, what kind of coloration and how the carapace and other body parts look like, and how color pattern varies from one part of the body to the other. Antennae are grayish brown, and the bryopods and pleopods are brown, and the rostrum of Penaeus monodon has 7 or 8 dorsal and 2 to 4, occasionally 5 ventral teeth. This is how Penaeus monodon looks like, and here using a diagram given in a very reputed research paper, I have tried to show you different parts of the Penaeus monodon body. Here, this is well labeled diagram. You can see that the terms which you will often uh, hear in the lecture about rostrum, about the telson, pleopod, uropod, preopod, and about the maxilliped antenniflagellum and antennal scale, antenniflagellum, and the hepatic spine. All these aspects are very important when you are discussing about the biology, and when we shall be discussing about the maturation and other stages, then you will find mention of many of these body parts. Habitat and behavior also we have discussed that they um, occupy different diverse habitats. In fact, in brackish, estuarine, and marine environments, we know that adults live in marine environments, whereas juveniles in brackish and estuarine. Depth at 110-meter, inhabiting bottom mud or sand. Monodon prefers to live at water temperature that we have already discussed, quite wide tolerance to temperature and salinity, especially salinity. And now the topics which we have not discussed in my previous lecture about feeding of different stages of Penaeus monodon. The food of Penaeus monodon consists mainly of crustacea, small crabs and shrimps and mollusks, making up 85% of ingested food. The remaining 15% consists of annelids and others. Um, Penaeus monodon is more of a predator of slow-moving benthic macroinvertebrates rather than a scavenger or opportunistic in feeding habit. So we know that Penaeus monodon feeds upon crustacea, especially crustacea, small crabs and shrimps, mollusks, annelids, and slow-moving benthic macroinvertebrates. In fact, it is not considered as benthic scavenger, but you know, as predator, slow-moving predator, you can say. The feeding habit appears to associated with the, if you will study its feeding behavior, then it can be associated with tidal phase. Shrimp nauplii is a non-feeding planktonic stage, right? It is a non-feeding and planktonic stage. It cannot move on its own. The three shrimp protozoa stages, then protozoa stages are non-selective filter feeders. Protozoa stages are filter feeders that feed mostly on, when they are filter feeders, that means they will feed on plankton, phytoplankton and zooplankton. So this is how different life stages of Penaeus monodon differ in feeding. From the mysis through to post larva, the PL stages, there is a transition to active predation on large zooplankton. Juvenile Penaeus monodon are considered to be carnivorous. They feed on a variety of items, in fact. They are carnivorous, but basically you can say that broadly they are omnivorous. They can feed on seaweed, algae, crustaceans, detritus, mollusks, and some of the fish parts if they get. Adults feed on crustaceans, annelids, algae, and detritus or mud part. It is quite difficult to determine the age of an individual prawn, but what people have studied, that lifespan of Penaeus monodon is about 2 years. Villaluz in 1969 described, and other authors also have studied the lifespan, but more or less similar information on its lifespan has been reported, like 1.5 years in case of males and about 2 years in case of females. That was reported by Moto in 1981. Then Mohammedan cited Srivatsa's work, who reported lifespan of prawn in the Gulf of Kutch to 12 to 14 months. So you can say that we can consider that prawn lifespan of prawn is 1 to 2 years in general of Penaeus monodon. Now, the males and females in case of Penaeus monodon are in different individuals. This is heterosexual. Female attains a relatively larger size than male. In the growth also, I mentioned that they attain total maximum size more than males. In fact, the genital opening on the coxa of the fifth periopod, I showed you there in the diagram that where periopods are. In case of males, the genital opening is on the coxa of fifth periopod. Here, in case of female, the genital opening on the coxa of third periopod. That is one of the distinctive features, that how you can differentiate between males and females. A joint petasma, a pair of appendix masculine on the exopods of the second peliopods. So, a joint petasma on the second peliopods. And in case of females, the reproductive structure in case of females, the thalicum is situated between the fourth and fifth periopod. So, these positions, these locations of these structures can help you better in identifying the sexes in case of Penaeus monodon. Male internal reproductive organ has paired testes, vasa differentia, and terminal ampoules. In case of females, they are represented by paired ovaries and oviducts. This is how the male and female openings look like. In case of males, you can see here the petasma is shown, showing the male specific structure. Here, thalicum is showing in the female Penaeus monodon. In this diagram, we have tried to show, this has again been taken from a source. If reference has not been provided, it may be given at the end of the slides or end of the lecture. Life cycle of Penaeus monodon, in fact, if you start from here, right from juvenile stage, adolescent stage, then sub-adult stage, adult stage, here depth has also been shown, and from when this adult stage spawns, then eggs, from the eggs, nuclei stages, different nuclei stages emerge. Then we have many protozoa stages, mysis, then megalopa or PL stages which are stocked for further rearing. So this is how the life cycle of monodon goes on, and different kind of environments that we have discussed in the beginning only, that they exploit estuarine, inner littoral as well as outer littoral zones to complete their life cycle in different phases of their life cycle. Now, eggs of Penaeus monodon are spherical, yellowish green, and are very minute. They are very minute because these are considered highly fecund. I will tell you later in the slides that they lay large number of eggs. Diameter is 0.27 to 0.31 mm. You can imagine that how minute, how tiny they are, with an average of 0.29 mm. Not even 1, but 0.29 mm. Eggs tend to sink slowly in still waters, and outer littoral area is the habitat where they lay eggs. Now, when cleavage starts, then two-celled stage can be reached in half an hour, four-celled in 1 hour, morula you can get in 1.8. Average time is shown here, and nuclei stages may appear in 11 hours. We all know that the developmental stages are strongly dependent on temperature variations and the climatic conditions. So when we start raising the temperature, then the developmental stages show faster pace. So that may vary from one condition to the other, but this is what on an average people have observed, researchers have observed. The nuclei in each egg is observed to move intermittently before hatching. There are different larval stages of in case of Penaeus monodon. Here you can see that we have 6 nuclei stages and 3 protozoa stages, 3 mysis stages, 3 or 4 megalopa substages, and their habitat is littoral area. Nuclei utilize yolk granules, we all know, within their body for getting their energy. Feeding starts in protozoa and mysis, collectively what we call as zoea stages. So feeding starts in case of Penaeus monodon from zoea stage, because the nuclei stage they have feeding from yolk granules. The protozoa and mysis, what we collectively know as zoea, protozoa 1 or zoea 1 has compound eyes under the carapace and are thus not clearly visible. Zoea 2 has a pair of stalked compound eyes, and zoea 3 has a pair of biramous uropods. So these are some of the features which you need to understand in order to identify different zoea stages. After 4 to 6 days, the protozoa finally metamorphoses into a mysis. Mysis shows antennal and thoracic propulsion. So this way you can differentiate between different zoea stages, and that is very necessary when you are going to get these for starting your culture practice. The megalopa stage, in fact, this is the PL stage, which is considered the seed which you need to stock for starting Penaeus monodon culture. Megalopa with the earlier juvenile stage traditionally called PL or fry for commercial purpose is transparent with dark brown streak on the ventral side tip of the antennal flagellum to the tip of the telson. This is very crucial. If you wish to start culture of Penaeus monodon, you need to understand that you will have to identify the seeds which are being supplied to you for culture. They should be PL stages. If you are not getting PL stages, the chances of mortality, the success of your culture may get adversely impacted. So be very particular in identifying the PL stages. Try to stock only PL stages, not below that. Under laboratory conditions, PL became benthic on the 6th day of PL stage, in natural conditions, these megalopa stages enter into the nursery ground, and the carapace length of megalopa varies between 1.2 to 2.3 mm. How to identify these PL stages or megalopa stages? Um, there is quite confusion sometimes in identifying the PL stages. So, to identify, we need to have a look, or we need to observe certain characteristics through which the identification becomes easier and unambiguous. Shape of the rostrum, that is the first point that you have to look into. Either straight or slightly bent upward at the tip. Rostrum should be either straight or slightly bent upward at the tip. Second, these PL stages usually have 5 dorsal spines but devoid of ventral spines. They usually may have 5 dorsal spines but do not have ventral spines. Inner or lower antennular flagellum is twice lower than the outer or upper flagellum. So, that flagellum you will have to observe that about upper and lower flagellum, how long they are and how they compare to each other. The sixth antennal spine does not have spinioles. These may help you, these points may help you out in identifying the PL stage which you wish to have in your pond. The chromatophores of dark brownish red extend from the tip of the inner antennular flagellum to the tip of the telson. When viewed microscopically, 13 chromatophores align ventrally along the sixth ears or are densely distributed to form an almost continuous pattern. The chromatophore on the anterolateral margin of the sixth ears is absent. So, those were the points which you can use for identifying the PL correctly. Then juvenile stages have transparent body, dark brown streak on the ventral side as in the megalopa, or what we popularly call as PL stages. Their habitat is usually estuarine area. How early juvenile stage and late juvenile stages compare to each other? In case of early juvenile stage, sixth abdominal segment is relatively shorter compared to the carapace length, and they have greater body size. Complete rostral spine formula you may get in early juvenile stages. Complete gill system is developed in early juvenile stages, and they are also benthic. Whereas late juvenile stage, body becomes blackish and bulky. 7 dorsal or 3 ventral spines in rostrum may be seen. Crawls using periopods, carapace length 2.2 to 11 mm. And in case of early juvenile stage, sixth abdominal segment is relatively shorter compared to carapace length. Here carapace length is 2.2 to 11 mm. Now, the next stage is adolescent and subadult stages. Adolescent stage starts resembling very much to the adult prawn. So, when you look at its adolescent stage, their looks are very close to how adult prawn looks like. Sexes become distinct in case of adolescent stages. Again, this is distributed in the estuarine area. This occupies estuarine area. Minimum size of males 30 mm carapace length, and minimum size of females 37. Remember that we talked about females being larger than males. Subadult stages. Here, in case of subadult stages, sexual maturity starts. Onset of sexual maturity. Male, in case of males, spermatozoa in its terminal ampules. While characterizing the male and female, we discussed about ampules. Then, in case of female, The thelycum contains spermatozoa. Spermatozoa from the ampules are transferred into thelycum. Females grow faster and migrates from nursery to spawning ground. Habitat littoral area, inner and outer littoral area. Minimum size of males in the subadult stage is now, they're slightly bigger. Males minimum size 37 millimeter and minimum size of females 47 millimeter carapace length. Adult stages, sexual maturity in adult stages is completed. Differs in size and habitat from subadult stages. Um, Habitat again offshore, inner or outer littoral area, minimum size of males 30 mm carapace length, and in case of females 37 mm of carapace length. In fact, this stage has appendages very similar to the subadult and is characterized by the completion of sexual maturity. It differs only with the subadult in size and habitat. Males possess spermatozoa and females start to spawn offshore, although a few spawn in shallow water. A second or more copulations may occur in majority of these species. Major habitat is the offshore area up to about 160-meter depth. Life history phases of the giant tiger prawn, Penaeus monodon. This table has, compilation of almost all the aspects of the life history. Here we can discuss one by one. Embryo stages, this is the phase which is given, and when it begins, what is the duration, what is the carapace length, CL, what we know, mode of life and habitat. So embryo first begins at fertilization, we all know, after fertilization, and then duration is 12 hours. Carapace length in case of male and female, when it is at embryonic stage, there is no distinction, so 0.29. Mode of life, planktonic, and they are in the littoral area, outer zone. Larvae, just after hatching, then duration 20 days, carapace length 0.5 to 2.2, again planktonic, and the same area, littoral area, outer as well as inner. Juvenile stages, when completion of gill system occurs. This table is very nice in this, describing the characteristics which develop with the different stages. 15 days duration, 2.2 to 11 millimeter is the carapace length. Now they become benthic. From juvenile stage, they become benthic and now start occupying estuarine area. Adolescent stage then comes, stability of body proportion, development of outer genitalia starts in the adolescent stage. In the previous slide also we discussed about this. Duration 4 months. Here onwards you will get separate information of carapace length in case of male and female. In case of males 11 to 30, in case of females 11 to 37 carapace length. Now onwards they will remain benthic. So the only stages which were not benthic, embryo and larvae. After that, from juvenile onwards, all stages are benthic and occupy this stage. Adolescent stage also occupies estuarine area. Subadult stages, it commences here, commencement of sexual maturity and first copulation in subadult stage, 4 months. 30 to 37 in case of males, carapace length, 37 to 47 carapace length in case of females, and they are also benthic and occupy inner and outer littoral area. Adult stages, completion of sexual maturity has occurred in adult stages, 10 months is the duration. Here in case of male, the carapace length is 37 to 71 mm, whereas in case of female 47 to 81 mm is the carapace length. They are benthic, outer littoral area. I think in the previous slide the CL of adult male and female was not clearly given. We can recheck. Adult 37 to 71 in case of adolescent, and adult stage. In case of adult stage, okay, this is the minimum size given. So it's all right. This is the minimum size, and uh, there we have discussed the range. So they can go to 71 carapace length, and here maybe 81. So accordingly, if you need to have corrections, I suggest you to please use this figure while describing the carapace length of different stages from this table, rather than giving minimum and then sometimes maximum, and that may be confusing. I suggest you to please take the data from this table while describing the carapace length of male and female. Now, spawning and fertilization. Spawning, we all know, refers to the release of eggs and spermatozoa um, by the two sexes of the prawn into the water for fertilization. The spermatophore, which contains the spermatozoa, is deposited in the female thelycum during copulation long before spawning, and from the ampules they are transferred into the thelycum. Penaeus monodon spawns 4 times in its lifespan. That can spawn 4 times in its lifespan, and probably has multiple spawning in a single season. Mating occurs at night, shortly after molting, while the cuticle is still soft. And sperm are subsequently kept in a spermatophore inserted inside the closed thelycum of the female. So that refers to the spawning behavior of the prawn in natural conditions. That what kind of conditions it requires, and how the process of spawning, breeding, and spawning occurs. So uh, molting can occur anytime, but more often during the night time. And these are all these steps are associated with spawning process in case of prawns. So that's it in this lecture, and in the next lecture we will talk details of culture practices and how Penaeus monodon is cultured. Thank you very much.